So first of all, do we know what DNA is made up of? There's one thing, I'm not sure, and try not to look it up. Yep, so molecules, yep, that's good. So it's not made up of blood, actually. Blood is something different. Well, blood on its own is a little bit different. But yes, DNA is found everywhere. I shouldn't say that. Anyway, it's made up of these things called nucleotides. So a nucleotide is what makes up the DNA. So that latter shape, all that latter shape is because it's made up of nucleotides, and nucleotides are made up of 3 different things. So think of it like we have our Earth, right? So Earth is our big planet, and you can think of Earth being made up of 3 main things. So you've got water, you've got land, and you've got air. So those are 3 things that Earth is made up of. So think about that. Yep, so try not to look it up too much, but I'll be saying what it is anyway. So those 3 things though, for DNA, since DNA is not made up of land, water, and air, they're made up of 3 different things called, there's a phosphate group, and then there's a sugar group, and then there's something called a nitrogenous base. So does anyone know the nitrogenous base? What is something that sounds really familiar? What do you think a nitrogenous base is made up of? Close. So what's maybe a word in nitrogenous that's really similar to stuff you can think of, maybe chemistry, similar to oxygen? Anyway, so nitrogenous bases are made up of nitrogen. So basically it's just a ring, and that ring has nitrogen in it. So it's an element in chemistry. So that is what a nitrogenous base is. And then somebody had said it before, but you guys are right, there's 4 different types of nitrogenous bases. So I know this is getting a little bit complicated, but DNA, I'll remind you, it's made up of nucleotides. Nucleotides are split into 3 different components: the sugar, the phosphate, and the base. Maybe we'll just call it a base. And then from the base, there's 4 different bases. And so those bases are what actually make up our, I'll call it our genetic code. So our genetic code is what will tell us to do things. So for example, we could have the 4 different bases, can be in any type of order that we want, and they are what talk to the cells, and they are what code for different things. So that's why I said code. So for example, if you have 1 different type of order of DNA, it could say, that could be the code for, maybe after this you should go outside and play, or play some sports, play some activities. Or you could have a different type of code that would, yep, so you're right, body traits. This is more body traits. Perfect. So the DNA is what encodes for everything. So that's what I will be showing you guys too. And the model that we're going to make today is you guys will be able to see how those 4 different bases can be super different, and they can be in a lot of different orders, and they can code for lots of different things. So does anybody know the names of the 4 different bases that we're going to talk about today? Yep, so somebody said it. So there's A, B, G, and C, which stand for, you guys don't need to know the names, don't worry, but I'll say them anyway. There's adenine, there's thymine, there's guanine, and then there's cysteine. So there's the 4 different types of bases that we're going to talk about, but for today, I'm just going to refer to them as A, G, C, and T. All right, so what happens in DNA is, in order for the latter, like we had talked about before, there's a specific shape that it needs to be, and there's only one way that it can fit in that particular shape. So what happens basically is A and G, 2 of the bases that we talked about, are bigger. So they're actually 2 rings stuck together. So those 2, so that's A and that's G. And then we've got C and T, and those rings are actually, or those bases are only 1 ring, so they're a little bit smaller. So this is something, like you guys had said earlier, so Watson, Crick, and Franklin together, they had kind of found this out, that the only way that DNA works, the only way that it sticks together and can encode for everything else, is if you have one of the bigger rings, so the ones that with 2 rings attached to it, is attached to just one of the ones with one ring, right? So that would be, so like I said, A and G are the bigger ones, A and T are the smaller ones. So for example, an A, which is one with two rings, will have to pair with T, which is one with just one ring. So that's the only way that they can fit together, is if you put a bigger one and a smaller one together, and those will attach, and there will be something called hydrogen bonds between them that keep them together. Don't worry about knowing that. That's just kind of something fun to note. So in terms of that, so that is what will form the DNA, and you'll have multiple pairs together. And does anybody know, between the DNA pairs, they're in pairs. So we have, like I said, there's A and G together, and then there's C. Do we know which ones pair together? So they have to be, there's a matchup for it to work. So what it is actually A and T go together. So those are the only two that can go together. And then there's G and C. Yeah. So close, even though I'd said, so remember with A and G, those two are both big ones, and C and T are the smaller ones. So you might think that they could pair up between each other and they could mix and match, but it's actually A and T are the ones that need to go together, and G and C are the ones that pair up together. So that's how they have to go. And then sometimes what will happen if they don't pair up right, so maybe let's just say that the A pairs up with a C instead. So that is something sometimes that we can refer to as a mutation, or something that will happen in the cells, and that's what happens. So that DNA would not be paired correctly. So sometimes that can result in really big problems happening, and other times, sometimes it doesn't do anything and it doesn't affect you in any way. But yes, and then in that case, I think we're going to move on to making our models. And then just so that you guys know, don't worry if you don't have everything right now or you can't keep up. This is being recorded, and we will post it later, so you can always come back and do it later. But if you guys do want to do it with me now, I will show you everything you guys need, and then we'll get started. So what we are going to use is, I have two black pipe cleaners here, so they're the full long ones, and you'll need two of it can be any color, it doesn't have to be black, just two pipe cleaners. And then what I'm going to do is I have 4 pipe cleaners of a different color, so you can see they're red. And then what I also have is I have 4 different colored, 4 different colored beads. This one is yellow, and then we have a blue right here, and then there is a green, and there is a white. So what I want you guys to do, it's okay if you're not doing this right now, but what you're going to need to do is you're going to need to pair 2 of the colors together. So for example, you can decide now that, so for me, I decided that blue and yellow are going to go together, and I also decided that white and green are going to go together. So you can also write it down if you want to think about it, or if you want to have a little bit more clarity for it. I wrote it down so we know, because A and T have to pair together. So I decided that A would be blue and T would be yellow. So that's why I said that A and T are going to pair up together, so blue and yellow. And I decided that white would be C and green would be G. So that's why I have white and green here, and those 2 guys are going to go together. So you want to make sure that those colors stay together throughout the whole time. All right, so once you guys have done that, you're going to take your 4, in my case I have red pipe cleaners, but you're going to take these guys and you're going to split them into 3. So you can either just take your scissors and you can just cut wherever you think 3 is, or yeah, so you guys can use, it doesn't matter how many you have. So when you actually make your DNA model, you can do, so for me, I have 6 colors for A and T, and then I have 4 different, or I have 4 beads each for G and C, which are the green and white ones. So that's the ones that I chose to do, but you guys can do it a little bit differently, or you can have more or less when you make your DNA models. It doesn't have to be exactly the same, but this is just what we're gonna That's what I'm doing right now. Anyway, so I decided I'm going to kind of fold this into 3 so that I know where to cut. So see here, there's kind of 2 indents, one here, and I'm going to cut those. And it might make a little bit of a mess sometimes with the pipe cleaners, but that is okay. So now you can see I cut my one big pipe cleaner into 3 smaller ones. So you're going to want to do that for all 4 or however many that you have. So now I'm just going to kind of go and cut it. Just because I did this earlier, so I know what it should look like into 3 pieces. And don't worry if they're not exactly the same size. You can use string. So yeah, there's a couple different things. If you guys don't have this stuff, actually, you could maybe use string, or if you don't have beads, sometimes what you can do if you want is you can cut out like little strips of different colored paper, and then just like wrap them in a circle, and then you can tape them together or glue them together. If you have maybe Play-Doh or something like that, or clay, you could also use that and attach it to the pipe cleaners or the string, whatever you have. So there's lots of different ways to do this. This is just the way that I found easiest, and I had the materials on hand. Anyway, and then what we're going to do is we're going to take our 2 really, really long pipe cleaners that I had before, and I'm just going to cut almost about, I'm going to take my index finger and I'm just going to kind of mark it from the bottom, and I'm just going to cut off just a little bit here because these ones are a little long. So I'm just going to cut that and make it a little bit shorter. You can just put the extra piece of the long pipe cleaner to the side. So for this one though, you are going to want to make sure that your pipe cleaners are the same size, or roughly the same size. Don't have to be exact, but as long as they're really close, then that should work. And then so for this part as well, this is what I was saying, how all these base pairs, so those nitrogenous bases that we were talking about, the 4 different ones, A, C, G, and T, they can pair up in lots of different ways. So you don't have to follow the order that I'm doing. But so for example, I'm going to take one of my small red pipe cleaners. I'm going to take the blue one. So for me, that was A, and I'm going to slide it through the pipe cleaner. And then I'm going to take the matching one, which is yellow, which goes for T because A and T pair together. And then I'm going to put them on the pipe cleaner like that. So you can put them closer together for now if you want, but we can move them around later. So I'm just going to do that. And then what I'm going to do, I'm just going to put it off to the side, and I'm going to fill up the rest of my, so I'm going to take another A and T, which is blue and yellow. It also doesn't matter. So you guys see here how I have blue and yellow in that order, but let's just say I wanted yellow on this side. So if I did that, and like so, and I did it like that instead, that is totally okay. As long as it is, I have A and T here that are paired together. It doesn't matter if it's this way or this way. It doesn't matter the order as long as they are together. That's what matters. So now I'm going to take a G and a C. So for me, that's my white and my green ones. I'm going to put that to the side. And so this is where you guys can just pair it however you want. You could do, if you wanted, you could do 6 A and T's in a row, and then you could have the 4 G and C's all together. So you can do it in whatever order you would like to do it in. So let's see here. So I did another A, or I did another G and C, sorry. And then now I am doing another A and T like that. And so sorry if I'm going a little fast for you guys. Like I said, there is going to be a recording posted, so you can go back and look after. And yellow, blue on the other pipe cleaner. Yeah, so for the colors, you guys can make them whatever you want. In this case, like I said, I had made my my G and C be green and white. But if you guys wanted to make your G and C be blue and yellow, or if you wanted to mix up, if you wanted to put, instead of the white with the green, if you wanted to put a white with a yellow, but you would just have to make sure that it was for the right matchup. So they just have to pair correctly. So once you're done all of those, so I'm going to do about 10 or so. So you got to get all of these guys together first, and then I'm going to show you guys how you can pair it up and make it look like DNA model. Like that. So there's my other one. Almost done here. And Also, like I said, you guys can make it however long or however short you like. So for me, the reason why I cut these black pieces or these black pipe cleaners originally is because I'm only doing about 10 or 12 of the smaller pieces, so that those are the ones that will make, these guys will make the ladder, so they'll go in between. So because I'm only doing about 10 of the smaller ones, I'm not going to need as much space on the long pipe cleaners. So that is why I cut these guys. But if you want to do a lot and a lot of these ladder pieces or the bridge pieces that go in between, then you can keep your pipe cleaner very long. So what we're going to do next is I will hold this up for you guys. Now I have all my different bridge pieces, so you can see here they're all there. And then what you're going to do, so you're going to take one of your long pipe cleaners, and I'm going to start with one side, and I'm going to pair this. So what I'm going to do is I'm going to take the tip of my pipe cleaner, and I'm going to wrap it around. So I'm just going to wrap it a few times, and try not to have it kind of sticking out like that. You want to try and get it so it's wrapped all the way around, just like that, and then straighten it out so it looks like this. So see, it's attached right there, and so you're going to want to do that, just like that. And so now you have a choice. You can either take a second pipe cleaner, and it can be, this is where it can be any order. It doesn't have to be a specific order. So I took another A and T and then another A and T. So you can either wrap it again around the side like this, or what you could do, if you guys wanted to, instead, you could take the other pipe cleaner along the side and you could start wrapping it like that, and then you could just go down like this. So now you guys can see this end is not attached, but this top one right here is attached on both ends. So this is starting our ladder. So it's starting to look like a ladder now. So for me, I'm just going to go down and I'm going to attach things, or the bridges on the side first. So with the nitrogenous bases, I'm going to go down one side and do it, and then I'm going to go down and do the other side. I can go slower for you guys if you want. Remember, this will be recorded too, so you can go back. So there's a third one, and what you guys will probably notice, you can see here, is they're starting to go up and they're going on an angle, and that is because the pipe cleaners are they like to do whatever they want to do, I guess I should say. So they like to kind of stick up in all different directions, but don't worry about it right now. If they're you can see this one's kind of going up there. As long as you have a similar distance between these guys, so you can see here how this distance in between these two is similar to this in between here. When we attach it in the end, it'll all be okay, and you can fix it around. So don't worry too much if these pipe cleaners kind of start going everywhere. All right, so now I'm going to take my next one. I'm going to wrap it around, and just as a reminder, see here how I've got my blue and yellow, and then a blue and yellow, a green and white, and then another blue and yellow again. And then this is a reminder that you guys can do whatever order you want. Just again, so this is what we were talking about, how our genetic code is different. So I decided to do this order, but one of you guys might have decided to do a green and white first, and then a blue and yellow, then maybe another green and white, and then another blue and yellow. So if you guys wanted to change it up, then that is totally okay. You can do whatever you want. So now you guys can really start to see how it kind of looks like a bit of a ladder. So this is a straight ladder. And remember what we had talked about earlier too, how it forms the ladder shape, but then what it'll do is it'll twist all together in the end, and it'll form something called a helix. So that's what we call it. So it's kind of it's like a spiral. So it will spiral together. So yes, at the end we're gonna take our DNA ladder, and I'm going to show you guys how to twist it so it will look like a twisted ladder, or a spiral, or in what we call a helix. So it'll look like that all at the end. So I will keep showing you guys what I'm doing here. So like I said, just taking the end and wrapping it around. And that's okay if your pipe cleaners start to kind of look wonky or they're not as straight anymore. That's okay. You can see here mine's doing some weird stuff. It's curving. That is totally okay. We're going to fix it all up at the end once we get our, I like to call them the bridges on, or the ladder pieces. So these to me are kind of the bridge pieces, the red ones. They're connecting the two pieces, the two black pipe cleaners together. So let's see here, wrapping that around, and then I only have 2 more left to go, and then I will show you guys how to connect it all. Yeah, so it's not going to be perfect, don't worry about that. Okay, so now I have finished one side. You guys can see the other side is not connected yet, so you're going to go through. And so this is where you kind of want to make it so that it stays straight, so you don't want to have a curved end. So for me, I'm just going to match it up with what's on top. So this guy here, it's about that length, so I want to make sure that the one below it is the same length, so my ladder in the end looks somewhat pretty. Doesn't have to be perfect though. And then I'll take the next one, go down, and then so I can move the black pipe cleaner kind of side to side so that it matches up with the length. Nice. So like I said, that's what I like to do. I wanted to do one side first, and then now I'm connecting it to the other side piece, or the black pipe cleaner, but you guys can do it. So the model, you can have the model in whatever order you want. Like I said, you have to keep it the same. So for me, and whatever colors you choose, my blue and yellow always go together, and my green and white always go together. So you need to make sure that those guys are always paired up, and those colors are not changing, but whatever order you want. If you wanted to put green and white here instead of a blue and yellow, you can totally do that. All right, so I'm gonna wrap this up. So I can show you guys here. So I'm wrapping it up again. And I've got these 3 left, so we're just gonna kind of twist that. And then you guys can kind of start to see how the ladder shape is really forming, right? This is a little bit of a twisty ladder, but that's okay. And so this is what I was saying, where you can make your ladder a lot longer. I decided to cut the black pieces and only do about 10 of the red bridges, but you can do however many you want. So I only pulled out enough beads to have about 10 ladder pieces, but that's okay. If you guys have a lot more beads that you want to use, or if you have a lot more pipe cleaners, you can make multiple different DNA models. So you can make like 5 different ones of these with all different genetic codes down the middle. That's okay too. Anyway, so this is my finished ladder piece. It is done. You guys can see it just like that. So we have our 2 black pipe cleaners, which, does anybody know what the black pipe cleaners are for the DNA model? It's called something specific. So the black pipe cleaners, those represent the DNA. It's called the backbone. So that kind of makes sense, right? It's the back of the DNA. It's what holds up the entire thing. So think of your back, right? Your back keeps you straight, it keeps you up. So that's the same with these, the DNA backbone. So in this case, it's the black pipe cleaners for me. Might be a different color for you, that's okay though. So that is the DNA backbone. And remember earlier what I was talking about with we have our nucleotides, and our nucleotides are made up of the 3 different parts, right? Like Earth. So we have our sugar group, we have our phosphate group, and we have all of our 4 bases. So we know that all of our 4 bases, these are the beads, right? Those are our 4 different bases, so we covered that. But where do you guys think the sugar group and the phosphate group are in the DNA model that we had talked about? Does anybody know? They make up something in our DNA model. So what the sugar group and the phosphate group, where you can find those are actually in the DNA backbone. So those are right here. So those are holding up, those alternate, so it goes a phosphate group, a sugar group, a phosphate group, a sugar group, a phosphate group, a sugar group. So that'll alternate, and that'll go all the way down the side of the backbone piece. So on each side, on this side and on this side. So that's where you guys can find the sugar and the phosphate group. I hope that kind of makes sense. And then, like I said, then we find our nitrogenous bases all throughout the middle. So those are the 3 different components of a nucleotide, right? And those nucleotides make up the whole entire DNA model, what we have right here. Okay, Next, what we're going to do is you have your ladder shape, and then the way that I'm going to do it, which I think works easiest, but all you're going to do is you twist it. So what you're going to do, I took 2 hands and you just twist it in opposite directions because you want to make it wants to be like a helix shape. So you guys see here how it's all twisty, like that. Yeah. So what I did again, here I can straighten it back out, and I'll show you guys one more time just in case you didn't see it. So I took my DNA model and then I put one hand on top, one hand on the bottom, and I just went opposite ways. So this hand went forward and this hand went backwards, and I just twisted it. And if you want, you can kind of just keep twisting it until you are satisfied with it. It doesn't have to be perfect. It's probably going to look a little bit wonky, but that's okay. So here's my twisted DNA model, and that is our helix shape. So it's like a spiral. You guys can see the spiral look to it. And you guys were right, it's our twisted ladder. So our twisted ladder, and we've got all of our components of the DNA built up together so that it stays like this, right? All right, and so do you guys know what are maybe some cool, we kind of talked about this before, but does anybody else know some cool, what we can do with DNA, or what it, whether it's in the medical field or what it can, so we know that it codes. We know that it codes for things and it codes our body. What does anybody know? Yep, so I saw family trees. So you're right. That is stuff like, if you guys have ever heard maybe about things like Ancestry.com on commercials when you're watching TV, stuff like that. So Ancestry.com, sometimes what they would do is that they can pretty much test out your DNA and then pair it with other people's who are similar. So if you were to take your DNA and your mom and dad's DNA, or your brother or sisters, if you have siblings, or maybe cousins or aunts and uncles, your DNA would be pretty similar. So actually, a fun fact about DNA is that 99% of all of our, or 99.9% of all of our DNA, so for me, and anybody else that we have here, anybody else that's here, 99.9% or percent of our DNA is going to be exactly the same, and only the 0.1% of the DNA that's all in our bodies is what makes us different. So that was just a cool thing that I found online. But going back, what can we use? We had talked about earlier, you can use DNA for crime labs. You can use it for things like, yes, Ancestry.com, so you can test out DNA. Does anybody else have any ideas as to what DNA could be used for? Why it's a pretty cool thing? Any suggestions? Anyway, so DNA with crime labs, it can be used for fingerprinting. It can be used, so if there's a crime scene, you're right, people can go to the crime scene, they can find things that might have DNA on it. What they'll do is they'll scan that DNA and they will match it. They'll put it in a database and see if they can match it up with anybody. So that's how they find sometimes criminals or people that were involved in maybe a crime scene. So that's kind of cool, and you might see that on TV if you watch any crime shows and stuff. It can also be used a lot in medicine. So we use it, we can test out DNA all the time to find out sometimes if people have a medical condition, or if they are sick, or if they have a genetic something, maybe a genetic disease or something like that. And you can test out your DNA and see if you have it, or maybe you can see if you're more prone to it, because sometimes, so for example, maybe this DNA model that I have can code for some type of genetic disease. So maybe this, or this gene code that I just made is really is found in a common genetic disease. So for that type of stuff, sometimes they can test your DNA if they find this, and if they find out that maybe you are more common for genetic disease, then they can watch out for that. So that's kind of cool how they can do. And every day they're still figuring out new ways to use DNA, and new ways to, something that they do is they amplify it. So that is something when you can take a really, really small amount of DNA and then you can put it in a big machine, and you can, it's called amplifying it. So it's making lots and lots and lots and lots of copies of that DNA. So along with that, we are actually going to talk about DNA replication. So that's when one strand might turn into two, and then those two strands will make more, so then it'll be four. Does anybody know why we might want to replicate our DNA, or why we want our cells to grow? So if DNA replicates, that also means that our cells are growing, right? Because it's important in something called cell division. So same type of thing, it's where you could have one cell and it turns into two cells, and then those two cells will turn into four more cells. So that's what we mean by amplifying it. So it's called DNA replication. So like I said, it's really, really important in cell division. So what we're going to do is I'm actually going to show you guys how it works. I have another DNA model here that I had made up. I made it up yesterday so that I could show you guys this and wouldn't have to make it again. So what's going to happen in DNA replication, like I talked about, it's really important so for example, think about you grow, right? As humans, we grow. So when you're a kid, you're a lot smaller, and then as you get older, you grow. So your legs will get longer, your arms will get longer, your feet will get bigger, your hands will get bigger, all that type of stuff, right? So those are all examples of reasons why, or those are all examples of cell division. So our cells are multiplying and they're making, let's say, our hands bigger, right? So my hand used to be maybe only the size of my palm, and then my cells were growing, my cells were expanding, and then because of that, also the DNA is expanding, the DNA is replicating, so now my hands are bigger, right? So that's an example. We cannot clone ourselves yet, no, but yes, that is a good point. They have found ways in some animals actually to take DNA. So they'll take a DNA model, they can take the entire genetic code. So genetic code, like I said, is all of this. So this is one kind of code, so this might code for something. They'll take the whole genetic code of an animal, like goats, and then they can replicate that. So that's a little bit more complicated, but it definitely is something that was pretty new and cool in the science world when they learned how to clone. I believe it was a sheep or a goat was the first animal. Anyway, really quickly, we're just going to go over how DNA replication works, and then we're going to do a few questions. So what happens is that my one DNA model here, you can think of this, so this will be in a cell, for example. What's going to happen is these bonds in between our nitrogenous bases right here are going to break. So I'm going to use scissors to do that. So there's something that comes in, a molecule that will come in, and it will break the bonds. So I'm going to use my scissors as that. So see here, I cut it, and the bonds are broken now. And then it's going to come along, and it's going to keep cutting just like that. So now our DNA ladder is going to break. It's starting to break into two separate strands, right? So it's splitting right down the middle, and all these bases are off to the side, just like that. It's going to keep going all the way until we have, let's just say, two separate strands. I'm not going to break off the top part yet, just so it's easier for me to hold it, but you can see here there's two different strands. So what's going to happen is so now I'm going to use my other DNA model that I had made. So another DNA model, but it's going to be a single strand. So we're going to want to cut this too. What's going to happen, I will show you guys, it's pretty cool. There will be another type of something really close to DNA that's only one strand. So see here how it's cut in half. And that is now going to come in. So this is a single strand. Now we have our DNA here, which is splitting apart, right? So we wanted to make this one DNA molecule, this one DNA, we wanted to split it into two. We wanted to divide it and replicate it. So what's going to happen is another strip. So this is something called RNA. So it's very similar to DNA, but it's a little bit different. So this is RNA, and this will come in, and it will attach to the other DNA molecule. So it'll attach like that. So you can pretend that these two guys are attached together. So it'll attach together like that, and then you could have another molecule. It's called RNA, but another strip that will come in, and that will attach to, let's just say, the other side of DNA. So see here, I don't know if you can see, but now we are essentially splitting our strand in two. So that's one side, it's split down the middle, and then we have another side here, which is splitting down the middle. So now you guys can kind of see we have two separate ends, right? We have two separate ends, and those are going to split off, they're going to break off, and we are going to form two brand new strands. I'll cut the top now. So now we have two brand new strands. There's one here, and there is one here together. We can pretend that those guys are together, just like that. So now we went from one strand to two strand. So that is how DNA replication works. So basically copies, so these two strands though would be exactly the same. So that's what happens when you copy DNA. It will copy the bottom line of it, and it'll make sure to match up. So remember we talked about those pairs, the A and T, the G and C that need to always be paired together. So it will make sure if this is my strand that I have that needs to be replicated, something will come and read it, and he could read this bottom guy and say, oh, that's a G molecule, right? So that's our guanine. So that needs to be paired with a C. So for example, something will come along, it'll read that, and then so it knows that a G needs to now pair up with this C. So that's something that DNA replication will do, and it will go down the whole DNA molecule and now form a brand new molecule. All right, so that is just a brief overview of DNA replication that I thought I would show you guys really quickly. I think it's something pretty cool that we can do, and so that's what I was talking about in the applications of DNA, where we can do something. We use it in the medical field all the time, so hospitals will use it or labs will use it, and they will replicate DNA. So they will make something, they can make one, if they only have one small piece of DNA, and then they can put it in this big machine that will replicate it, and you could go from one piece of DNA to like 450 pieces of DNA, for example. So then that makes it a lot easier to study the DNA and see what the DNA is composed of. So that is all for today. I am going to open it up for a question period. So if you guys have any questions about any of that, I know that was a lot of information and a lot of stuff going on, but like I said, it will be recorded, so you guys can go back later, we'll post it, and if I went too fast with the model, making the model, you guys can go back and look at it and see what I did for that. So if you guys have any questions, you can go on and you can either ask it in the chat or you can turn on your mic.